For AP biology or college biology, we're going to be talking about Unit 2, cell structure and function, membrane transport. Please note the following disclaimer below. Let's take a look at the following picture that's listed here to help you understand the difference between transports and their main purposes. Firstly, I want to draw a picture of a cell. And remember, a cell has a plasma membrane, which has all these different organelles inside, and the plasma membrane helps protect the cell and also is sort of like a door to allow only certain things to come in and out. With the fluid mosaic model, we learned that they're embedded with proteins and they are different types of proteins, and that is what the plasma membrane has. We also have glycolipids and glycoproteins, but altogether, the plasma membrane works to protect the cell and only allow certain things to come in and out. Now, the model also, remember, talks about selective permeability, and that's what is the term used to describe only letting certain things in and out. And that is done with different types of transport, hence the table here or graph here. So with this table, graph, mind map, I should really say, I decided to help you understand the basic gist of what we're talking on a higher level. Firstly, we're talking about the difference between passive versus active transport. And in this further video, we're going to be taking a look at the nitty-gritties of the specific types of passive versus active transport. So, first of all, the main difference between passive and active transport is active requires some sort of energy, and the energy is known as ATP. The way that I like to remember it is A. So, active transport begins with the letter A, and so does ATP. The other thing to note is that with active transport, it moves against the concentration gradient versus passive transport moves along the concentration gradient. The way that I want you to think about it is if you have a treadmill in an airport, for example, and if you've ever been to the airport, sometimes they have this treadmill thing that allows you to move with the movement. So, what I mean by that is the treadmill will move in one direction, and you want to get from point A to point B. So, you get on that treadmill and you move along with it so you can move faster. That's the same way passive transport works. It moves along the concentration gradient where you're moving in the same direction as that treadmill in the airport. Now, active transport is sort of like you on a walking pad or you on a treadmill in your home, where you are standing in one place and you're on this treadmill at home and the treadmill moves in this direction and you are moving in this direction. So you are moving against the concentration gradient. And that's another way that I like to think of active transport is like being on a treadmill where you're moving against the concentration gradient and therefore you require a lot of energy versus if you were using the one in the airport, it's allowing you to move faster and you don't really have to exert that much energy. So that's the difference between active and passive transport and the understanding that passive does not have energy and active does. Now, there's different types of passive and active transport that we're going to be talking about. I want to draw your attention to osmosis. You could see the little arrow here. And the thing is, the reason why I drew that arrow is that osmosis has its own proteins. And so sometimes it's considered to be under the topic of facilitated diffusion, but in most textbooks, they say that it's not. It's just an active transport, or should I say passive transport. So because of this, I just have it in a tiny subcategory on its own. So it might be confusing with this, and we'll explain it more in detail in just a second. Okay, so now let's just focus on passive transport and the two main giant categories. So remember, passive transport moves along the concentration gradient. It is like you on that airport treadmill, just no energy required. And there are two main categories of this, and that is diffusion, or sometimes known as simple diffusion, and then facilitated diffusion. So when they talk about simple diffusion or just regular diffusion, what they're talking about is moving different particles or molecules from areas of high concentration to areas of low concentration, but we don't have any sort of protein that is there. It's just an easy flow through, and that's simple diffusion. Whereas facilitated diffusion also goes from areas of high concentration to low concentration. However, it requires some sort of protein. So it can have this sort of, you want to think of being on the highway and having a bridge that you're going under, and you have this opening to allow for movement. So that's facilitated diffusion. There is some sort of transport protein that helps assist it. Now let's talk about osmosis, which is a category in of itself. Now, some might say that it is facilitated diffusion in the sense that it uses proteins, but this is not always the case. Osmosis is its own subcategory, which is why this arrow sort of is in the air. It's sort of in between. Basically, it is a type of passive transport, and that's what you need to know overall. So when they talk about osmosis, we're talking about the movement of water through a semi-permeable membrane of some sort. And the proteins that we're talking about with osmosis are called aquaporins. And so that's where it might fall under facilitated diffusion. However, when we're looking at the way that it moves, we're talking about the direction of water movement to move from areas of low solute to high solute concentration. And this is where it gets a tad bit awkward. And it's used to maintain the cell turgor, which means the cell sort of strength in a sense. So, when we're talking about osmosis, I want you to think about these two images here because they love to give free response questions or multiple choice questions with osmosis using these two objects, either some sort of glassware of some sort, and then sometimes you'll see a U tube, like literally it's called a U tube. And the way that they describe it is there's some sort of membrane, which can be seen here or here, and it'll say in the question, hey, I gotta move some water. There's some sort of water movement, and it can only move water. That's the barrier that it has. So you know they're talking about osmosis, and they'll let you know that there is a higher concentration of some sort of solute on the one side compared to the other side. And so to make it even, what ends up happening is the water will move to the area of the higher solute concentration. Say, for example, if there's a higher salt concentration on this right side here, it will literally move the water from the left to the right to allow for water movement so the concentration becomes even in a sense. So that's osmosis. Now let's talk about active transport. Remember, active transport requires some sort of energy. So when you're looking at pictures related to the phospholipid bilayer and you're trying to determine whether or not it's passive or active, you're looking for ATP as your way of knowing that there's energy, and therefore it must be active transport. When it's active transport, we're moving against the concentration gradient. So remember, it is like walking on a treadmill where you're exerting energy and you're going against the grain, essentially. Now, there are three main categories of active transport. So a sodium potassium pump, endocytosis, or exocytosis as the three main examples. Now, with sodium potassium pump, you're not really required to know that much about the pump, except if you do see some sort of protein and you see that there's some Na and K on one side versus the other, they're just trying to move the Na and K through a pump, which they utilize some sort of protein. They show some sort of protein that looks like a pump, and they're using ATP to move those ions across the barrier. We don't have that many details that we need to know about that for AP biology, but that's the brief overview in case they give you some sort of question related to that. So now we have endocytosis versus exocytosis are the two other forms. Endocytosis is when you're importing materials into the cell, and exocytosis is when you're importing materials outside of the cell. And the way that you can remember that with ease is you think enter, exit. So enter begins with en, and so does endocytosis, and exit begins with ex, and so does exocytosis. So you can think of endocytosis, enter, import in, and exocytosis, exit, ex. Now, with endocytosis, it could be categorized into three main categories as well: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So if you look at this picture here, if you have phagocytosis, you're engulfing some sort of large particle. So you can think of phago meaning big, and pino, you want to think like pint or tiny. You're engulfing tiny particles. Usually both phagocytosis and pinocytosis are essentially creating a vesicle or vacuole of some sort to integrate into the cell. And then when you have receptor-mediated endocytosis, which will come up in differentiation in later units, you have to think that the cell has these little keyholes, which are called receptors or ligands that exist. So you have these receptors, and what ends up happening is the receptor will attach to the key or the ligand, which will open up the door. So let me explain that again. So you have receptor-mediated endocytosis. The cell itself is lined with these receptors, and what ends up happening is you have a key essentially that will open it, which is the ligand. And then what ends up happening is it ends up creating this vesicle that allows for that enclosure, which the receptors are now inside with their ligands itself and basically are like a coated vesicle. So that is receptor-mediated endocytosis. It allows items to go into the cell with the assistance of the connection between the receptor and the ligand. So now that we've covered that part, let's try to answer a quiz question. Let's take a moment and read the following question. Please note that for this question, you're wanting to take a moment, pause the video, read the question, and then come back once you've selected an answer. Okay, now that you're done selecting an answer and you've unpaused the video, let's go over this question together. The question states, assume the cell is permeable to only water or water only. If the cell contains 0.5 m solution of NaCl and the environment contains 0.2 m solution of NaCl, which of the following statements are true? So which of the following statements would be true, it essentially says. And they give you four options. A, the cell would decrease in volume. B, the cell will burst due to osmosis. C, there would be a net movement of water into the cell. Or D, there would be no movement of water at all, basically. So the way that I like to think of this question is drawing a picture to help assist. So we are talking about a cell of some sort, and they're telling us that there's 0.5 NaCl on the inside and 0.2 NaCl on the outside. And it doesn't matter, the solute or the NaCl or salt is not going to move. It's only going to be the water that moves. So because the cell is permeable to only water, we know that they're talking about osmosis. Now, when we have osmosis, that means that only water can move. So I'm going to skip down to answer choice D, where it says there would be no net movement of water. This is an extreme answer with no as well. And remember, that's not really favored because we know that osmosis can move water only, but can still move water. So the answer choice D is not favorable. Now, going back to A, it says the cell will decrease in volume. We're not talking about any specific type of cell, and we don't know this for fact based on the information that's provided to us. So because of this, A is not favorable either. Now, we're going to talk about the ways that cells can decrease in volume in a later section, but for now, we're just talking about a basic cell, and you would know this because they didn't specify a cell wall or any sort of fact related to that, which we'll cover once again in a later section. But for now, you just need to know we aren't given that information. We aren't told any information related to that. So it's not really favorable, especially compared to answer choice B and C. B says the cell will burst due to osmosis. We haven't talked about bursting, but again, that's in a later subsection. However, osmosis doesn't really cause bursting for this specific problem, so it isn't really favorable. And then C, it says there would be a net movement of water into the cell. And so that would make sense that there's some sort of movement because of the different concentrations. Whether or not that's higher or lower, we don't know. So because of that, we are just going to state that that's C. We don't know if there's additional items, but that's the best answer choice compared to all the answer choices. Remember, for AP Bio, you're picking the better answer of all of the answers. So we're basically saying C is the best answer. We know that later on we're going to talk about hypertonic, hypotonic, but for now, you just know that based on the answer choices you were given, that C is the better option of the four. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't wait to help you succeed.